Hi, welcome to this talk on COVID-19 vaccine development. My name is M Ndosi, a researcher and an academic in the west of England. I will give you an overview of COVID-19 pandemic, why vaccine is important, the process and the landscape of vaccine development. We all know that COVID-19 is a highly infectious disease. It is caused by a newly discovered coronavirus. The technical name here is Severe Acute Respiratory Syndrome Coronavirus 2. The disease has spread rapidly throughout the world. Over 5 million people have been affected. Over 300,000 have lost their lives. This scenario is likely to be an underestimate because it relates to only confirmed numbers reported to the World Health Organization. The actual numbers are likely to be higher. Then we come to the reproduction number, the R number. This describes how many people each infected person will infect on average. For COVID-19, the reproduction number is 2.6. That means one person can potentially pass the virus to 3 people on average. 1 out of 3 infected people will have no symptoms, but they can still pass the virus to others. Therefore, the infection rate grows rapidly in the community. Recent research has shown that our number for COVID-19 is 5.7. That means one person with COVID-19 can potentially pass the virus to about 6 people. This is higher than what researchers originally thought. But to put this in perspective, measles has a reproduction number of 15. Therefore, measles is much more infectious than COVID-19. Now, most people infected with COVID-19, about 80%, will have no symptoms, or they will experience a mild illness and will recover without needing any special treatment. Some, however, will become severely unwell and will be admitted to hospital. A smaller proportion, about 5%, can be described as having a critical disease. These will often need breathing and other organ support in the intensive care units. Those who are more likely to develop serious illness are older people and those with underlying medical conditions, such as chronic lung disease, severe heart conditions, diabetes, kidney disease, cancer, and those with immune deficiencies. Why is vaccination important? COVID-19 has no cure. There are currently no treatments against the virus itself. The main treatment for patients with COVID-19 is supportive care. The aim is to maintain the functions of the body, keeping the individual alive while the disease progresses and eventually resolves itself. There are tests going on with medicines that are used for other conditions to see if they will work for COVID-19. Also, serum or plasma obtained from people who have recovered from COVID-19 is being tested as it contains antibodies against the virus. This could offer passive immunity, but this is only short term, and all these treatments are only experimental at the moment. COVID-19 has affected every area of our lives. There's been a lot of deaths at a rate that we've never seen before. Bereavement, suffering, unemployment, mental health issues, you name it. Primary prevention measures such as isolation, shielding, and lockdowns work. These help to reduce that reproduction number to under 1. However, lockdowns are not sustainable. What we need is that community immunity. This happens when so many people in a community become immune to an infectious disease that it stops the disease from spreading. This can be achieved either naturally or by mass vaccination. For COVID-19, that will mean letting about 60% of the population get coronavirus. But we know that a proportion of those people with COVID-19 will develop a critical illness and potentially die. Therefore, this may not be the best way of achieving that herd immunity. Vaccination, however, has been shown to be effective against the other diseases such as measles, which is even more infectious than COVID-19. This looks like the best approach to protect everyone, including those with immune deficiencies. We don't have vaccines for COVID-19 at the moment, but they are in development. Vaccines work by exposing the body to an antigen, which will not cause the disease but will stimulate the immune system to produce antibodies that will block or kill the virus if the person gets infected. There are different types of vaccines: live vaccines, dead vaccines, and others. Live vaccines use a weakened form of the virus that causes the disease, and because they are so similar to the natural infection, they will create a very strong immune response. A single vaccine injection, for example, is usually sufficient to induce long-term or even lifelong protection. Examples of these are measles, mumps and rubella vaccine, chickenpox vaccine, yellow fever vaccine, and others. However, we cannot give these live vaccines to people with weak immune system, such as those with underlying chronic conditions, the elderly, and yet these are the very people that are at greatest risk for COVID-19. These are the very people that we want to protect. Dead vaccines are viruses that are rendered inactive. They are unable to infect, but they can cause an immune response. They are safe. However, they do not evoke a strong immune response. Repeated doses or boosters are often required. Examples of these are whooping cough vaccines, rabies vaccines, hepatitis A vaccines, etc. And there are this group of other vaccines in development, protein subunits. These contain only a protein part of the virus that is responsible for evoking an immune response against COVID-19. And then DNA and RNA vaccines. These work by introducing genetic material coded for COVID-19 spike proteins, which are the antigen. Okay, and once injected into the body, the cells use the genetic information to produce COVID-19 spike proteins. And this will be detected by the immune system, therefore evoke an immune response. And then there's this clever method, viral vector method. This uses a virus that does not usually affect humans, and the virus is genetically modified and given a gene for coronavirus spike protein. So when this virus is introduced into the body, it directs the cell to make coronavirus spike proteins, which are displayed on the surface of those cells and therefore recognized by immune system and triggering an immune response. Clever way, really fantastic research going on there. But there's no guarantee that all this will work. Developing new medicines for treating diseases is a long and complex process involving a thorough scientific and regulatory process. This ensures that medicines are safe and effective. Now, vaccines are not treating any diseases, they're aimed at healthy individuals. Therefore, they need to be super safe. There are two stages to vaccine development: preclinical and clinical development. Preclinical development is the research done in the lab and on animals. It involves testing on artificial environment. The steps start with the identification of the antigen. That's the molecule that induces the immune response in the body, down to the manufacturing of the vaccine. The clinical development involves testing in humans. This is to ensure that the vaccine is safe and effective in the natural disease conditions. Clinical trials require ethical approvals, and this involves scrutiny by independent ethical committees to ensure that the research follows strict ethical principles, including informed consent and doing no harm. There are at least 3 phases to clinical trials, and sometimes a 4th phase may be required to assess effects of long-term use of the vaccine. Phase 1 clinical trials are usually small-scale studies on healthy volunteers. Researchers will be asking the questions here: is this vaccine safe in humans? What sort of immune response does it evoke? If this phase is successful, then the research will continue to phase 2 clinical trials. Again, these are usually small scale, and the question is, does it work against artificial infection? What is the effective dose? And again, looking at the side effects and the sort of immune response this vaccine will generate. So if this phase is successful, the research will progress to phase 3 trials. These are usually large-scale, multi-center studies. They're usually randomized. And the research question is, does it work under natural disease conditions? This is the definition of effectiveness. Again, we'll assess safety over time. And if the drug or the vaccine is shown to be safe and effective, then the developers can apply for approvals. Depending on the countries, these boards are usually independent. They will look at all the data, all the safety data, all the effectiveness data, and if they're happy and they satisfy themselves with the process, then they will give approvals. Then this vaccine can be used for humans or can be marketed. In clinical trials, we usually have phase 4 studies, where these are called post-approval surveillance. Okay, the aim here is to assess the effectiveness or the effects in different populations, or to see whether there are any rare side effects or adverse effects. Are there side effects associated with long-term use? We've seen drugs that have been in use and later on withdrawn from the market because of these phase 4 surveillance studies. So it is a very robust and rigorous process. So this is the COVID-19 vaccine development landscape. At the time of preparing this talk, there were 118 different projects of vaccine development. 8 of those are in clinical evaluation stage. And it's wonderful to see different countries being involved, from Australia to the United States of America. There are at least 8 types being tried against coronavirus. Some are virus-based, some are viral vector, protein-based, nucleic acid-based, and a few 3 unknown. A lot of these projects are driven by the private sector and a small proportion by the academic, public, and not-for-profit organizations. So this talk has given you an overview of the pandemic, why vaccine is important, the process and the landscape of vaccine development. So the virus is here, but there are reasons to be hopeful. We are developing that community immunity against coronavirus. Vaccine development is an example of good science, science for the common good. But we must remember that science answers with a degree of uncertainty. For example, vaccines may not work as expected. Also, science is limited. It may not be able to answer all your questions. Science cannot weigh up the trade-offs that people have to make. Some may decide not to take the vaccines, for example, and for different reasons. However, science can be a powerful tool to help us make good decisions. If you need more information on vaccine development, please follow the links below. I am on Twitter, and you are welcome to follow me if you wish. It is a good platform for discussion, which is always good for advancing our knowledge. Thank you.